Hello dear students, welcome to a lecture on human pedigree symbols and construction of pedigree, inheritance pattern, presentation of molecular genetic data and risk assessment. The main objectives of today's lecture are: number first, to understand precisely the human genetics. Number second, to have precise discussion on human pedigree symbols. Number third, to help students understand how to perform pedigree analysis and how to assign an inheritance pattern. Number fourth, to have an overview about molecular pedigree analysis. And finally, to have a brief discussion on risk assessment. Dear students, let us first of all start with the human genetics. Human genetics is the study of inheritance in human beings. It encompasses a variety of overlapping fields including classical genetics, cytogenetics, molecular genetics, biochemical genetics, genomics, population genetics, developmental genetics, clinical genetics, and genetic counseling. A human pedigree is a graphical representation of the pattern of inheritance of a particular trait, usually a disease, in a family tree. Pedigree analysis is a principal tool in medical genetics for predicting whether a newborn child will or will not suffer from a genetic disease. However, molecular pedigree analysis is the use of molecular data to understand pedigree, and included in this are all the methods that use molecular data to test conjectured relationships between the individuals. In addition to the testing of specified relationships, for example, parent-offspring relationships, we take molecular pedigree analysis to include estimation of the level of relatedness between individuals, and it is useful for the prediction of heritable disease risk. In modern medical genetics, pedigree analysis is typically combined with tools such as polymorphism analysis via PCR and electrophoresis of the DNA of patients and their relatives in order to make an accurate diagnosis. Now, let us discuss pedigree symbols. A pedigree is a diagram showing genetic information from a family using standardized symbols. The frequently used symbols in the human genograms are: number first, proband, that is, an affected family member coming to medical attention independent of other family members. Possession of the character under study is shown by a solid or blackened symbol, and absence is shown by an open or clear symbol. Next is male. The male is represented by a square on the left of the family tree. Then comes female. The female is represented by a circle on the right of the family tree. Next are offsprings. In a standard genogram, there are three different types of children, that is, biological or natural child, adopted child, and foster child. Next comes marriage. The offspring symbols appear from left to right in the order of birth and are connected to the marriage line by a vertical line. Then comes relationships. A break in a relationship line, that is horizontal line, indicates the relationship no longer exists. Multiple previous partners do not need to be shown if they do not affect genetic assessment. Then comes consanguinity. If degree of relationship is not obvious from the pedigree, it should be stated, for example, third cousins, above relationship line. Then comes carrier. It may not likely manifest the disease regardless of inheritance pattern. However, asymptomatic or pre-symptomatic carrier is the clinically unaffected individual at the time of pedigree analysis but could later exhibit symptoms. Dear students, although we have discussed most of common pedigree symbols, but still there are several other symbols used in human genograms. Moving ahead, let us have a brief comprehension about important rules and conventions to remember while studying pedigrees. Number first, males are always represented by squares and females by circles. Number second, an empty circle or square indicates that the individual does not suffer from the disease. Number third, a shaded square or circle indicates that the individual does suffer from the disease. Number 4, a square or circle with a line through it indicates a deceased individual. Number 5, an arrow pointing to a square or circle, that is the proband, indicates the individual who is under consideration, that is usually the person who first seeks medical attention. Number 6, a horizontal line between a male and a female indicates a mating pair. Number 7, a double horizontal line between a male and a female indicates an incestuous mating pair, that is between two people who are second cousins or closer. Number 8, the male should always be drawn left of the female in a mating pair. Number 9, a vertical line that leads from a horizontal mating line represents the connection between parents and their children. Number 10, children are always represented in order of birth, that is the eldest being the farthest left. Number 11, individuals in a pedigree are denoted first by generation, that is in Roman numerals, and then in order of their appearance horizontally in Arabic numerals. Now, moving ahead, let us discuss how to perform pedigree analysis. Genetic pedigree analysis is concerned more and more with the search for genes that play a role in very complicated disease pathways. For complex diseases, it is the case that a single gene may act in concert with additional genes or environmental exposures. Pedigree analysis includes these attributes, that is locus, heterogeneity, penetrance, phenotypic variation, and the presence of phenocopies. Human pedigree is usually performed for the reasons: number 1, to define whether an experimental trait is indeed inherited or environmentally determined. Number 2, to explain the mode of inheritance of an allele of a given gene, that is dominant or recessive, or autosomal or sex-linked. Number 3, once the mode is understood, to counsel parents on the probability or risk of passing on a given trait. Number 4, as gene hunting technique, for instance in some newly discovered trait inherited along with some known characteristics. Now, let us discuss the steps involved in human pedigree analysis. Number 1, assigning disease status for pedigree members is accomplished first by dropping the set of blank founder chromosomes through the family, with each parental chromosome equally likely to be transmitted. Number 2, the proband might have received a segment that originated from a founder and a segment from his or her married-in parent. Number 3, given that the proband is affected, the disease genotype is assigned in accordance with specified disease allele frequencies and penetrances. All pedigree members with the same founder chromosome as the proband will receive the corresponding disease allele. The remaining founder chromosomes are assigned a disease allele consistent with given disease allele frequencies. At this point, all of the people in the pedigree have a disease genotype assigned. Affection status is then determined based on penetrances. Marker genotypes are assigned to all individuals starting with the founders. Two markers, for example A and B, each with two alleles, for example 1 and 2, would have 4 possible allele combinations or haplotypes. For each haplotype, 2 conditional frequencies must be designated, one in the presence of the disease allele and one in the absence of the disease allele. Inheritance pattern. Once a pedigree has been constructed, the principles of Mendelian inheritance are used to follow the trait through a family to determine whether it is inherited as a dominant or recessive trait and whether it is located on an autosome or a sex chromosome. The 5 basic Mendelian patterns of inheritance for traits controlled by single genes are: number 1, autosomal recessive inheritance; number 2, autosomal dominant inheritance; number 3, X-linked dominant inheritance; number 4, X-linked recessive inheritance; and number 5, Y-linked inheritance. If you are analyzing a pedigree, first try to rule out all patterns of inheritance that are inconsistent with the pedigree. For example, only males carry a Y chromosome, and for traits controlled by a gene on the Y, only males will be affected. If the pedigree shows affected females, Y-linked inheritance can be ruled out. Second, if only one pattern of inheritance is supported by the information in the pedigree, it is accepted as the pattern of inheritance for the trait being examined. However, because of the relatively small size of human families, the pedigree may not provide enough information to rule out all but one possible pattern of inheritance. For example, the information in the pedigree may indicate that both autosomal dominant and X-linked dominant inheritance are possible explanations. If this is the case, the next step is to determine whether one pattern of transmission is more likely than the other. If so, if one pattern is as likely as the other, the only conclusion from the information available is that the trait can be explained by autosomal dominant or X-linked dominant inheritance, and that more work is necessary to identify the pattern of inheritance. This may require adding more family members to the pedigree or analyzing pedigrees from other families with the same trait. There are other problems that often complicate pedigree analysis, including variation in gene expression, the degree of phenotypic expression called as penetrance, and traits controlled by alleles that are common in the population. Now, before moving to molecular pedigree analysis, let us briefly discuss penetrance and expressivity. The terms penetrance and expressivity define two different aspects of phenotypic variation. Penetrance is the probability that a disease phenotype will be present when the disease genotype is present. For example, if all individuals carrying the allele for a dominant disorder have the mutant phenotype, the gene has 100% penetrance. If only 25% of those who carry the mutant allele show the mutant phenotype, penetrance is 25%. If the phenotype of a trait is present in less than 100% of those with the related genotype, the trait is said to show incomplete penetrance, while as expressivity refers to the degree of a gene's phenotypic expression. Expressivity defines the degree of expression for a particular trait. If a trait does not have a uniform level of expression, it is said to have a variable expressivity. This variable expressivity results from interactions with alleles of other genes and with non-genetic environmental factors. Dear students, now let us discuss the new approaches to pedigree analysis, that is, molecular pedigree analysis. Molecular testing offers advantages that it can be performed on any tissue source of DNA and does not require sampling of affected tissue. This means that diagnosis can be performed on blood or fetal samples, for example, chronic villus or amniotic fluid samples, obviating the need for invasive biopsy of tissues such as liver, kidney, muscle, brain, and so on, and also permitting prenatal diagnosis. It also can be done regardless of whether an individual is symptomatic, allowing for early diagnosis of individuals at risk of inheriting a genetic condition. The first application of molecular diagnosis involved linkage-based testing rather than mutation analysis. This was the case when genes involved in particular disorders had been mapped but not yet identified. Linkage is still used in instances where it is difficult to identify pathogenic mutations, either because the gene is unknown or because of the difficulty in finding mutations in large genes where a wide diversity of mutations may occur. Dear students, molecular pedigree genetic data presentation can be discussed under these headings. Number first, restriction fragment length polymorphism or RFLP, and number second, genome-wide association studies, that is GWAS. Now let us discuss restriction fragment length polymorphism. Genetic markers are alleles that can be used to keep track of a particular gene in an individual or population. The marker is not the disease gene itself, rather it is a recognizable allele that segregates with the disease allele because they are very tightly linked on the same chromosome and they tend to be inherited together. Restriction fragment length polymorphism have become the key element to diagnose diseases. A particular RFLP is usually associated with a particular genetic disease, then the presence or absence of that RFLP can be used to counsel people about their risk of developing or transmitting the disease. The assumption is that the gene we are really interested in is located so close to the RFLP that the presence of the RFLP can serve as a surrogate for the disease gene itself. Let us look at how these experiments are performed. First, as with any genetic comparison, we need variable phenotypes and some way to detect them. Human diseases can be detected clinically. Sickle cell anemia patients can be detected by the shape of their blood cells, and hemophiliacs can be identified by the inability of their blood to coagulate properly. Dear students, for RFLP analysis, a toolkit is restriction enzymes and nucleic acid probe. First, we need a restriction enzyme that will cut DNA or RNA in a manner that is informative for the disease in question. And second, we need a nucleic acid probe that will detect that region. Not all restriction enzyme sites will reside in the same position in all individuals. Thus, if we cut DNA with a restriction enzyme, we may not obtain the same size DNA fragments. For instance, screening for the sickle cell gene. Sickle cell disease is a genetic disorder in which both genes in the patient encode the amino acid valine in the 6th position of the beta chain of the hemoglobin molecule. Normal beta chains, that is beta A, have glutamic acid at this position. The only difference between the two genes is the substitution of a T for an A in the middle position of codon 6. This converts a GAG codon to a GTG codon and abolishes a sequence recognized and cut by one of the restriction enzymes. When the normal gene is digested with the enzyme and the fragment is separated by electrophoresis, the probe binds to a short fragment between the red arrows. However, the enzyme cannot cut the sickle cell gene at this site, so the probe attaches to a much larger fragment between the blue arrows. Both the father and mother are heterozygous, that is, semi-filled box and circle respectively, as they had to be to produce an afflicted child that is represented by a solid box. The electrophoresis patterns for each member of the family are placed directly beneath them. Note that the two homozygous children, that is 1 and 3, have only a single band, but these are more intense because there is twice as much DNA in them. By testing the DNA of prospective parents, their genotype can be determined and their odds of producing an afflicted child can be determined. In the case of sickle cell disease, if both parents are heterozygous for the genes, there is a 1 in 4 chances that they will produce a child with the disease. Amniocentesis and chronic villus sampling make it possible to apply the same techniques to the DNA of a fetus early in pregnancy. The parents can learn whether the unborn child will be free of the disease or not. The mutations that cause most human genetic diseases are more varied than the single mutation associated with sickle cell disease. Over 1,000 different mutations in the cystic fibrosis gene can cause the disease. A probe for one will probably fail to identify a second. A mixture of probes, one for each of the more common mutations, can be used. But there remains the problem of false negatives, that is, people who are falsely told that they do not carry a mutant gene. There are many diseases which result from several mutant genes working together to produce the disease phenotype. There are still genetic diseases for which no gene has yet been discovered. Until the gene can be located, cloned and sequenced, no probe can be made to detect it directly. Now, screening for an RFLP marker. The probe will hybridize, that is, bind to different lengths of digested DNA in different people depending on where the enzyme cutting sites are which each person has inherited. Thus, a large variety of alleles will be present in the population. Some people will be homozygous and will be represented by a single band, and others will be heterozygous with each allele producing its band. Second is genome-wide association studies. The common disease or common variant hypothesis holds that alleles at relatively high frequencies, that is greater than 1%, represent a significant proportion of susceptibility alleles for common disease. Their high frequency implies that association studies in large population cohorts will be fruitful for identifying risk alleles. Genome-wide association studies have successfully identified thousands of loci for a range of human complex traits and diseases. A single SNP may cause a Mendelian disease, though for complex diseases, SNPs do not usually function individually, rather they work in coordination with other SNPs to manifest a disease condition, as has been seen in Mendelian and complex traits later being characterized by mutations or variants in different genes and with often additive effects and environmental interactions. One approach to the study of genetic factors in common disorders relies on large population studies, looking for gene variants that are associated with an increased risk of disease. In its simplest form, this is done using the case control study. A set of individuals who display the phenotype under study, for example cases, are compared with a group who are matched as closely as possible, but who do not have the phenotype that is controls. Genetic studies are then done to determine whether there is significant difference in the frequency of a genetic trait between cases and controls. An odds ratio calculation is done comparing the frequency of the trait in cases versus controls. An odds ratio greater than 1 indicates that the genetic variant is positively associated with a disorder, and a value less than 1 indicates a protective association. Now, next comes risk assessment. Most common medical conditions, for example, coronary artery disease, diabetes and hypertension, are multifactorial in etiology. Multifactorial traits are determined by a combination of genetic and non-genetic factors. Evidence that genes contribute to a trait includes familial clustering, monozygotic twin concordance, and studies of heritability. Risk assessment or predictive test is based on genetic association, that is, looking for genetic variants that are associated with an increased risk of disease are not diagnostic tests. The test result will indicate relative risk for a disorder, not whether the disorder is currently present or someday will be present. Relative risk of disease in individuals of a specified genotype can differ in different populations, as can the frequency of any specific allele. Next comes HapMap. The HapMap is a catalog of common genetic variants available in the public domain, developed to help researchers identify genes associated with human diseases. The information contained in the HapMap is derived from the DNA of 270 individuals of African, Asian and European ancestry, which is the product of a multinational collaboration, that is, the International HapMap project. The catalog describes what the variants are, where they reside in the human genome, and what the frequency of specific variants is in certain populations. The HapMap takes advantage of the fact that genetic variants that are located close to each other are often inherited together. The ability to study common haplotypes instead of single SNPs allows researchers to more easily narrow the search for genes associated with specific medical conditions. Next is heritability. Phenotypic variation for a quantitative trait is attributable to the summed effects of genetic and environmental influences together with any covariances and interactions. The proportion of phenotypic variance contributed by genetic variation is termed the heritability. The heritability scale is the influence of genetic and environmental factors on phenotypic variation. This provides us with insights into the genetic and environmental architecture of human complex traits and our potential ability to dissect out loci associated with trait variation, and is also useful for the prediction of heritable disease risk. There are many lines of evidence that support the contribution of genes towards the determination of multifactorial traits. Geneticists measure familial clustering with a variable lambda, which is the ratio of the frequency of the trait in relatives divided by its frequency in the general population. This can be easily measured by looking at the frequency of the trait among siblings as compared with the general population, in which case the variable is lambda s. For an autosomal recessive trait, the frequency in sibling is expected to be 1/4, and for an autosomal dominant it will be 1/2. The value of lambda s will depend on the population frequency of the disorder. For the autosomal recessive disorder, that is cystic fibrosis in the Northern European population, lambdas is 0.25 divided by 0.004, that is equal to 625. And for the dominant disorder, that is Huntington's disease, lambdas is 0.5 divided by 0.001, that is equal to 500. Hope you might have enjoyed today's lecture. Thank you very much and goodbye.